Welcome to Poddy, March 23, 2026. A deep dive. Today, we're diving into what might be the most significant breakthrough in computational biology, EVO2, a biological foundation model that's basically learning to read and write DNA like it's a language. We're talking about a model trained on 9 trillion DNA base pairs from across all domains of life, and it's apparently the biggest biological AI model ever released. Plus, we'll look at why some experts are saying genomic language models could transform medicine, but not quite yet. So let's break down what this actually means and why it matters. Okay, so let's start with the big picture here. According to the headlines I've been seeing, researchers announced EVO2 inches February 2025, so this is very recent stuff. And the scale of this thing is just mind-blowing. We're talking about a genome language model that was trained on over 128,000 genomes, encompassing over 9.3 trillion DNA base pairs. To put that in perspective, that's like teaching an AI to read the entire genetic library of life on Earth. The model can apparently interpret and generate DNA sequences at whole genome scale while maintaining single nucleotide resolution, which is incredibly precise. Now, what's really fascinating about this approach is that they're treating DNA like a language, which, when you think about it, makes total sense. DNA is literally a code with 4 letters, A, T, G, and C, that combine to create all the complexity of life. So researchers are using the same transformer architecture that powers ChatGPT and other large language models. But instead of learning English or Python, it's learning the language of genetics. And apparently there's something called the EVO designer interface that's powered by this EVO2 model, which sounds like it could let people actually design genetic sequences. But here's where it gets really interesting. And this is my take based on what I'm seeing. This isn't just about understanding existing DNA. The headlines are talking about generating DNA sequences, which means we're potentially looking at AI that can write new genetic code from scratch. Think about the implications there. We could be talking about designing new proteins, creating synthetic organisms, or even developing entirely new biological systems that don't exist in nature. One headline literally says, "Biggest ever AI biology model writes DNA on demand." That's both incredibly exciting and, honestly, a little terrifying when you think about it. Now, there's also some interesting context here about other DNA language models that have been in development. I'm seeing references to something called Grover, which is described as a DNA language model that learns sequence context. And there's another one called Merge DNA that uses a hierarchical autoencoder-style transformer to compress and reconstruct DNA sequences. So EVO2 isn't happening in a vacuum. There's clearly been a lot of work building up to this moment. What's different about EVO2 seems to be the sheer scale and the fact that it spans all domains of life, not just specific organisms. But let's talk about the elephant in the room here, the computational requirements. The headline mentions that the computing power needed to process all this data makes EVO2 the biggest biological AI model yet released. We're talking about processing 9 trillion base pairs of genetic information. That's an enormous amount of data. And it probably requires massive computational resources that most research labs just don't have access to. This could create a real divide in the field between institutions that can afford to run these models and those that can't. And this brings us to a really important point that one of the headlines makes. Genomic language models could transform medicine, but not yet. That not yet is crucial. While the potential here is absolutely massive, we could be looking at personalized medicine, better drug discovery, understanding genetic diseases in ways we never could before. We're still in the early stages. The models might be able to read and write genetic code, but translating that into actual medical applications is a whole different challenge. You need clinical trials, regulatory approval, and a deep understanding of how these generated sequences actually behave in living systems. What's particularly interesting to me is the whole genome scale capability while maintaining single nucleotide resolution. That's like being able to see both the forest and every individual leaf at the same time. Most previous approaches had to choose either you could look at big picture genomic patterns or you could focus on individual genetic variations. But doing both simultaneously was computationally impossible. If EVO2 can really do this, it represents a fundamental breakthrough in how we can analyze genetic information. Now, I want to touch on something that's been fascinating me about this whole field, the connection to the broader trend of long context models in AI. There's been this push toward models that can process million token sequences, and that's exactly what you need for genomic analysis. A human genome has about 3 billion base pairs, so you need models that can hold massive amounts of context to understand how different parts of the genome interact with each other. It's like the difference between reading individual sentences versus understanding an entire novel. The context matters enormously. But here's what I think is the real game changer. If these models can actually generate functional DNA sequences, we're looking at a completely new era of synthetic biology. Instead of just modifying existing organisms, we could potentially design biological systems from the ground up. Want a bacteria that eats plastic? Design it. Need a protein that performs a specific function? Write the code for it. Of course, that's assuming the generated sequences actually work in practice, which is still a huge question mark. But even if we're years away from that level of capability, the foundation is being laid right now. So, where does this leave us? My take is that we're witnessing the birth of a new field, computational biology that can actually create biology, not just analyze it. EVO2 represents a massive step forward in scale and capability, but we're still in the very early stages of understanding what these models can really do. The potential applications in medicine, agriculture, environmental cleanup, and even space exploration are staggering. But we need to be realistic about timelines and challenges. This is cutting-edge research that will take years to translate into real-world applications. If you're curious about this stuff, I'd recommend keeping an eye on the publications coming out of the teams working on these models. This field is moving incredibly fast right now.